Okay, now we're going to go on to Chapter 2, but first we're going to do some review questions from Chapter 1. The first question, spontaneous generation is a theory that states A. Life comes from smaller life forms, B. Life comes from inanimate, non-living matter, or C. Life can be too small to see at the earliest stages. The answer is B. Spontaneous generation is a theory that states life comes from inanimate and non-living matter. The next question, microbes. A. Cause disease. B. Prevent disease. C. Both cause and prevent disease. The answer is microbes can both cause and prevent disease. That's C. Which of the following scientists is associated with the germ theory of disease? Is it A. Pasteur, B. Jenner, C. Reddy, D. Koch, E. Leeuwenhoek? The answer is D. Germ theory of disease is associated with Koch. There are two taxonomic categories that are present in every type of domain system. That's the 3-domain, the 2-domain, and the 4-domain. These include the A. Bacteria, B. Archaea, C. Eukaryotes, D. Both A and B, or E. Both B and C. The answer is D. Both bacteria and archaea, A and B, are in every domain system. The 3-domain, which has the bacteria, archaea, and eukaryotes. The 2-domain that has bacteria and archaea. And the 4-domain that has bacteria, archaea, eukaryotes, and viruses. So the answer is D, both A and B. Viruses are cells that can't reproduce, acellular obligate parasites, they live in extreme environments, or D, they're small bacteria. Viruses are acellular obligate parasites. B. Viruses are B. They can reproduce, they just have to have a host cell in order to do that. So you can't say A is the answer. They don't live in extreme environments. That's more archaea. And they are not small bacteria. So viruses are acellular obligate parasites. Okay, let's go on to Chapter 2. How we see the invisible world. Microscopy. These are the microscopic units of measurement that you will need to know. The first one is the micrometer or the micron or micrometer. You might hear it all kinds of different ways. We can visualize 10 to the negative 6 meters and 10 to the negative 3rd millimeters with our Brightfield compound microscopes that we have in the Mount Hood Community College lab. We can visualize down to this level. Electron microscopes can see at the nanometer level. That's 10 to the negative 9th meter and 10 to the negative 6 millimeter. That's what you need to know for units of measurement involving microscopes. Robert Hooke in 1665 observed cork cells, and he liked to look at plants because he liked their geometric shapes. And he developed a set of lenses that he put together, and he was able to observe the different shapes of cork. And he came up with the word cells. This discovery of cells marked the beginning of the cell theory, that all living things are composed of cells. The gentleman who developed the first known microscope is Leeuwenhoek. He made the first observations of living cells. He took cells from his own body and looked at them under a series of lenses that he put together. He developed this very first simple microscope. In his case, it only had one lens. He was able to have a specimen platform, and he was able to use a screw to adjust it. But this is the example of the first microscope. Compound microscopes are the type of microscope that we have in the lab at Mount Hood Community College. They are Brightfield compound microscopes. And we have two lenses that we can use. We have the ocular lens and the objective lens. The image from the objective lens is magnified through this second lens called the ocular lens. Total magnification is the objective lens times the ocular lens, and you need to know this mathematical equation. The following is a very important microscopic term that you need to know. It's resolution. Resolution is the ability of the lenses to distinguish two distinct points, to see fine detail clearly. The idea is that shorter wavelengths of light can provide the greater resolution. And the best way to improve resolution is through immersion oil or filtering out non-blue light or using electrons. Those all can increase resolution. If you look at the image on the far left, you see just one dot, a little bit of fuzziness around it. Then you get better resolution in the middle, and you can start to see that, oh, it's not just one dot. And then on the far right, you can see that there are two dots. That is the best resolution. That's where you can see two distinct points and clearly see the fine detail. These are two images. We use the light microscope, and you can see the image is a little bit fuzzy. But if you look at an electron microscope, which has increased resolution, you are able to see much more detail. On this slide, what you need to know is the refractive index. The refractive index is the light bending ability of a medium. You want the light to be able to come to a pinpoint. And sometimes the light may bend in air and it misses where it's supposed to be going, and you have to find a way to narrow down this refractive index. Immersion oil is often used to keep the light from bending out and escaping. It has the same refractive index as glass, so it's like you put glass upon glass, and so it increases the refractive index, and it increases resolution. You want to have excellent resolution. So again, immersion oil is the way to go for this, to make sure that you have it coming to a pinpoint and not escaping out the sides. Blue light also increases resolution. When you put in a blue filter, you can help balance out those red parts. If you look at the image on the left, you can see it has a lot of yellows, a lot of reds. But if you look at the image on the right that uses a blue light filter, then you can see it more clearly. So a blue light filter increases resolution. Brightfield illumination is what we have with our Brightfield compound microscopes. The dark objects or specimens that you look at are visible against a bright background. The light reflects off the specimen and it does not go into the objective lens. If you look, your eye is above the ocular lens. You look through the ocular lens, which goes to the objective lens, which goes down to the specimen. And using Brightfield illumination, you can see that dark specimen against a white or bright field. In Darkfield illumination, the light objects are visible against a dark background. You can see the object or the specimen a little bit clearer when it's against a dark background using Darkfield illumination. The light is reflected off the specimen. Phase contrast and differential interference contrast accentuate the diffraction, which allows for internal structure examination. If you look on the far left, you have the Brightfield illumination, and you can kind of see the specimen. In the middle, you have that Darkfield, which brings it a little bit clearer. And then using contrast, you are able to now see the little bit of cilia on that specimen. So contrast microscopy can accentuate the diffraction and allow for this visualization of internal structures. Fluorescent microscopy uses UV light and fluorescent dyes. They're called fluorochromes. These absorb the UV light and then they emit visible light when you use this type of microscopy. You can see in this picture how bright it is and how the fluorochromes have come to life and really lit up the slide. Here are some more examples of the fluorescent microscopy. It makes it really bright. You've probably heard of fluorescent dyes. Same thing, fluorescent stains. It makes them very bright and very colorful. In confocal microscopy, we are going to look deep within the structure of what we are observing. And we're going to use fluorochromes and a scanning laser. And then we are going to excite those fluorochromes so we can see deep within the structure and be able to visualize internal structures within that specimen. The photons illuminate the planes of a specimen. You have two-photo illumination, which allows the cell activity to be observed in real time. When the laser illuminates each plane in a specimen, it can combine them to form a 2D or a 3D image. Confocal microscopy is for thick specimens, and we again use this so that we can visualize deep within the specimen so we can look at the internal structures. The very highest resolution microscopy is electron microscopy and atomic microscopy. Atomic would be the highest resolution of all, but electron microscopy is also excellent for resolution. Electron microscopy uses short wavelength electron beams instead of light to increase the resolution and the magnification. We are going to look at two types in the following slides: transmission electron microscopy, also called TEM, and scanning electron microscopy, also called SEM. There is also scanning tunneling probe and atomic force microscopy that we will look at. And again, this has the highest resolution. It doesn't use electrons, it uses atoms. So first, let's look at transmission electron microscopy. The electrons are transmitted through a substance which has been sliced very, very thin and then stained with heavy metals. The key for transmission electron microscopy is that it uses these very thin slices so that you can observe the internal structures. This is an example of an electron microscope, and these are some images that it produces. The images are black and white. You might have seen some really colorful electron microscopy images, but those have been photoshopped. The color has been added to them. It makes them look like they're glowing, like a fluorescent microscopy image might show you. But in actuality, all electron microscopy images are black and white. So you can see this really colorful image right here that they have photoshopped, but in reality, it's black and white. Scanning electron microscopy causes the electrons to bounce off the surface of the specimen. It gives you a 3D surface image. The electron gun fires a beam of electrons and it bounces off of the specimen, but you can only visualize the outside. You can only visualize the surface in the scanning electron microscopy. It still produces a 3D image, but remember, it's just the surface. Here are some examples of scanning electron microscopy. Again, the colored ones have been photoshopped. They've just added color to the images to make it look really cool. In actuality, they're black and white images, but they are really nice looking, and they do show the surface of the specimen and produce a 3D image. This is at the atomic level for microscopy. The first we're going to look at is the scanning tunneling probe. It uses a metal probe to scan along the specimen at the resolution of 1/100th of an atom. This is the highest resolution. So the metal probe will drag across the specimen, kind of like how a record player needle would drag across the record. It scans and runs along the surface of the specimen. These are some examples of what a scanning tunneling probe image would look like. And this is at the atomic level. So the probe, the metal probe, has come along the outside of this image and produced that visible surface. Can you guess what this is? This is the first image of DNA, and it was produced using an atomic microscope. Before they were able to kind of guess and kind of piece together what they thought DNA looked like, but this is an exact image of it. This is another atomic microscopy image. What this is apple pectin. Have you ever heard the phrase, an apple a day keeps the doctor away? Well, it's could be true. What apple pectin can do is it can circle around a microbe and it can make like this little nest and kind of hold it tight. This pectin can encircle it and keep it in a tight little clump so that the immune system can recognize it as something foreign and get rid of it for you. So this is an image of apple pectin. Continuing with atomic force microscopy, you can also use a diamond probe. A diamond probe is much better because it will last longer. It's less likely to dull quickly, which a metal probe could dull, and you may not know that it's dull, and you may not get an accurate image. But a diamond probe can last a long time, and the image will remain crisp, and the resolution will be high. It again can produce a 3D image, and it is of the surface of what it has been moved across. CLASI-FISH is a relatively new type of microscopy. What it can do is give you like a snapshot of the environment of the sample that you have collected. It is not a pure culture. It is the diversity of all the microbes in a natural type of sample. In your mouth, you have over 700 different type of microorganisms that are present. And there can be numerous microorganisms present in a biofilm. And what CLASI-FISH can do is give you this very colorful image of all the different types of microbes that are present so that you can see the overall diversity of that sample. Now, the Mordants is a really cool microscope that is made out of paper. It was developed so that it could be used in places where there is no electricity, how you can use it out in a developing country because you want to investigate an infection that's present in a population, but you have no electricity. And so this paper origami microscope was developed to be able to use in places where there was no electricity. It's really interesting, and there is a TED Talk on the Mordants and the gentleman who made this. And if you're interested, there's a link at the bottom of this page. Let's review. Which unit of measurement is the smallest? A, millimeter, B, micron, C, nanometer. The answer is C, nanometer. So an electron microscope, remember, can see at the nanometer level. That's the size of viruses. We are not able to see that in our compound Brightfield microscopes that we have in the lab. We see at the micron level. That would be like for bacteria. So the answer for the smallest is C, nanometer. What is the total magnification for a microscope with a 10x ocular lens and a 40x objective lens? A, 40, B, 100, C, 400, D, 4,000. The answer is C, 400. What you do is you take the ocular lens number and you multiply it times the objective lens number. The ocular lens will always be 10, but the objective lens number will change. You take that ocular lens and multiply it by the objective lens, 10 times 40, and you get 400. The answer is C. Which term describes the clarity of an image? Being able to see two distinct points, the clarity, the crispness. Is it A, resolution, B, contrast, C, refraction? The answer is A, resolution. Which type of microscopy uses the thinly sliced specimens? Is it A, SEM, B, TEM, C, scanning probe? Thinly sliced specimens are from TEM, transmission electron microscopy. That is TEM, thinly sliced specimens, B. SEM was the scanning electron microscopy, and that was for the surface of an image. And remember, scanning probe, those were atomic level microscopy images. So for this answer, it was B. Thinly sliced specimens are TEM. Let's talk about specimen preparation for light microscopy. So the first thing you're going to do is sterilize your loop until it's red hot. And then you're going to let it cool for 30 seconds. After it's cool, you're going to put the loop into the test tube of sterile water and kind of scoop up a loop of it. It will kind of settle into the loop. And then you're going to tap that loop onto your slide that you are preparing, and that drop of sterile water will come off onto the slide. Then you're going to flame your loop until it's red hot, and you're going to let it cool for 30 seconds. Then you're going to take a little sample from the bacteria tube that you'll be using. Just a little bit, you don't need a whole lot. And then you're going to rub that loop full of sample into the sterile water, and you kind of smear it around into that water drop. Then you let it completely dry. After it's completely dry, you will pass it through the Bunsen burner flame just a couple of times, and this will heat fix the slide. This will kill the microbes and help them to attach to the slide so it doesn't wash off in the rinsing, the staining, the decolorizing, through all of the procedures that you are going to do with that slide. Unfortunately, heating the slide and helping to heat fix the microbes to the slide kind of distorts them a little, and so it isn't the absolute true morphology, because it's a little bit distorted. Contrast is very important to be able to see a specimen. Unfortunately, bacteria that's unstained is nearly transparent, so you have to stain it to create contrast so your eyes will be able to see it when you're looking through the microscope. Contrast results from the differences in the index of refraction between the specimen and the background, or within the specimen and the number of visible colors in the specimen. For example, black versus white would have the highest contrast. You would absolutely be able to see it. You can see the written words on this white background. That's the highest contrast. When we stain bacteria, we are creating a contrast so that our eyes can see it. We are now going to talk about stains. We will go more in depth about stains as the course expands and as we go along. But this is just a brief overview of the different types of stains that we are going to look at. First, we have the simple stain. It is easy, it's simple. The stain colors the specimen all the same color. We use a simple stain to look at morphology and arrangement. That's all we use it for. We're not looking for anything specific about the specimen, but we are able to see the morphology, which is the shape and the arrangement. In a differential stain, we are looking for something specific, something different about our specimen, about the culture that we have made. And in a differential stain, you will have different chemicals that you use in order to stain different parts of the bacteria or different types of bacteria, different colors. Some examples of a differential stain would be a gram stain. In the gram stain, we're looking to see if it's gram negative or gram positive, and we will go into this stain a lot. Acid-fast stains are to determine if they're from the family of Mycobacterium. These Mycobacterium have a special substance in their cell wall, and it's called mycolic acid. And we use the acid-fast stain because it helps us to determine if our specimen is an acid-fast type of bacteria that could be a pathogenic mycobacterium. Now, for special stains, we are enhancing special structures that we are looking for. We want to know if our bacteria has a capsule, or if it has an endospore or a flagella. These are special stains that we use to examine all the unique structures that a bacteria or our specimen might have. And we will go more into all of these different types of stains in the upcoming chapters. So again, that simple stain is using one single stain, one single type of dye, and we are staining everything on that slide the same color. The sample will all be one color, but you can see the morphology and the arrangements when you do a simple stain. A mordant will be used when we do the gram stain. The mordant we use is iodine, and it helps to hold the stain in that bacteria. It helps to coat the specimen. The mordant enhances the properties so that the color is enhanced. The mordant, because it kind of builds up on or in the specimen, sometimes can make the specimen to appear larger than it really is. This is an excellent table from your book, and it talks about the basic stains, the simple stains, it talks about acidic stains, and it talks about negative stains. And we will do both the simple stain and the negative stain in our labs. This is another table from your book, and it talks about the different types of differential stains. The gram stain, the acid-fast stain, the endospore stain, and the capsule stain are all stains that we can do in our lab at Mount Hood Community College. One of the most important stains that you will learn about is the gram stain. There are two types of bacteria that you will see in the gram stain. It will either be gram-positive or gram-negative bacteria. The gram-positive bacteria will stain purple, and the gram-negative bacteria will stain red. The reason why this stain works is because of the properties of the cell wall of the bacteria. Gram-positive bacteria have multiple layers of peptidoglycan in their cell wall. When you use crystal violet and the mordant, the iodine, in this stain, it forms a complex that sticks inside that very, very thick cell wall. When you decolorize this bacteria that is gram-positive, it will not wash off the purple color. If it's gram-negative bacteria, it only has a very thin layer of peptidoglycan in its cell wall, and it has these lipid membranes. When you decolorize a gram-negative bacteria, it pokes holes in that lipid membrane, and it helps to wash out that purple color from the crystal violet. It essentially makes it a clear bacteria. Because that crystal violet has not formed a complex, there's not enough peptidoglycan in that cell wall, and because the decolorizer has poked holes in the membranes, all the color washes away. So you counterstain this now clear cell with safranin, and when you're done with that process, it shows up red. So gram-negative bacteria will be red. Gram-positive bacteria will be purple. Another stain we're going to talk about is the acid-fast stain. It's a differential stain for pathogenic mycobacterium, like TB or leprosy. An acid-fast positive cell has a very waxy, sticky material inside of its cell walls called mycolic acid. This mycolic acid is very hard for the stain to penetrate. And once it does, though, it stays there and it is not washed out when you decolorize with acid alcohol. You leave on that primary stain for 5 minutes so that it can penetrate that waxy material. It is a long stain and it does take a while, but the results are really cool. The non-acid fast cells will lose the stain when they're rinsed with acid alcohol because they don't have that waxy material in their cell walls to hold that stain in. And so when you counter stain with your next stain, you'll have a completely different color. An endospore stain is another very important stain. Some bacteria are able to produce endospores, which are stress-resistant structures that can surround their genetic material so that they can retain it when they're under stress, when it's too hot, when there aren't enough nutrients for them to survive. They're able to produce endospores to protect their material and kind of wait until the conditions are better for them. So we can do an endospore stain in order to see if our bacillus has an endospore. And we use heat to force the bacillus to produce these endospores. Having an endospore can indicate pathogenicity. When we talk a lot about foodborne illnesses, we will talk about endospores. So remember the endospore for the future. The capsule stain is a type of negative stain. Do you remember the nigrosin that we used in the other negative stain? Well, this is going to have the same type of theory, the same type of procedure. We're going to stain the background around this bacteria and try and visualize whether this bacteria has a halo around it, which would represent a capsule or a slime layer. A capsule or slime layer increases the virulence for that bacteria and increases the pathogenicity of that bacteria. So the capsule stain will stain that background and it will repel against that bacteria, so it does only stain the background. And you will use a counter stain to stain the inside of that bacteria. There's no heat fixation, just like in the previous negative stain that we did. So there's no distortion of that bacteria, and you'll be able to see the true morphology and the arrangement. Here are two images representing a capsule stain. The image, especially on the right-hand side, really clearly shows that halo that's around that bacillus. So that bacillus would have a capsule around it. And when you look at the image on the left, the A, you can see how that background is very, very dark against that bacteria, and that would be a negative stain. And that's the end of chapter 2.